Sorry.
Okay, let's have a class. So, first of all, let's review what we did last on Tuesday. Okay, we can also go back to see what we did like last Thursday. Let's look at the urine sample. So I would be very interested to see is our two patients what it looks like. So we have two patients in the classroom. Are they safe? We want to see.
I think so.
My name's Beau.
Patient number 1 and patient number 2. So let's see. So first of all, what did you guys the strip test? So remember we had a strip test of the urine sample. What we are looking for is blood cell, is blood, is we call it a white blood cell, and we look at the last one is nitrite. So that's a 3 markers, the first thing. One of them are positive, it will be presumptively or suspicious urinary tract infection. Then we look at the results on the first 2 agar, which is a McConkey agar and a blood agar. Did you see their patient? They're good? Are they good? Patient number 1, blood agar is very clear. McConkey agar is very clear. And usually we don't need to do that, because if this is negative, we don't need to do that, okay? But anyway we did. This is an environmental contaminated sample. You see there's a yellow colony there, so that's good. So the second stage, we move on to McConkey agar. Then we move on to blood agar. That is what we call is qualitative test, and then we use a neutron agar to see the real number, and the real number it should be less than 100,000 CFU per ml, and this is the clinical standards. That's patient number 1 looks good. Number 2, blood agar looks good. I usually look at the blood agar first because it's enrichment media, everything will grow. McConkey agar looks good. And also, if you look at the neutron agar, that's very good. So there's nothing happens for these patients in the lab. They are very good. Shouldn't have a problem. Okay. Now let's look at what we have for our mated urine sample. So everybody look at your blood agar first. If you have the patient looks like that, that looks terrible, then you're gonna be face light to look what type of the colony looks like. Is there beta hemolytic? Probably not. If it's a beta hemolytic, that is even worse because it's going to be translocated from the kidney, which means it's invasive. So on the blood agar, we're also looking for whether there is a beta hemolytic. If there is a beta hemolytic, which means it is very invasive. That's an invasive pathogen which is coming from the kidney. Okay, then you look at your McConkey agar. What do you see? If you look at your McConkey agar, did you see it's a pink color? And the colony is very good looks. So what's a pink color tells us? If you see this is a pink colony, what this means, it possibly lactose fermentation positive. And of course on the blood agar, it is gram-negative bacteria. So that's suspicious, it could be E. coli. Then we look at this agar bites. Some of you are countable, some of you uncountable. I would say this is more than 100. How about that sample? Looks more than 100. This is a very good example. Let's see a couple of the other samples, what it looks like. This looks like, I don't need to see this one. This looks like huge too. That's more than 100. Let's look at that sample. That's more than 100 also. That is more than 100. More than 100. That's also more than 100. So all of you guys have the sample, which is clearly that's more than 100. Okay. Oh, that's good. We're going to talk about modes. So, this is what we did for our UTI test, the last lab. Okay, what we're going to do today? I said we had a bacillus, we also had a E. coli. We did on the milk, skim milk agar, split blue agar, and also the starch agar. Did you guys see anything different on the skim milk agar? Pretty much not. But I can tell you can record the results supposed to be Bacillus subtilis is all positive on one side. But what we get to know is this is really good. The plates we saw, there is a mode there. And it's at room temperature. So we're going to do some modes today. Okay, that's really good. That's what you have. Is the fact sheet chapter 14. That's the first thing we're going to do. Now, by the way, today we will do an introduction of the molded bread, and also we have this sample. That's really nice. We can say it's a lab sample. On Thursday, Amy will give a special lab talk detail about the fungi, and also in her slides, the last page will be a question in the exam for the extra points. Okay, so make sure you're attending and you're listening carefully. It will be a fun lab, because I'm not a specialist of fungi, and she's a specialist of fungi, and we talk about the bacteria all the time, and we should cover the fungi in the lab. So we'll have an introduction today, and we're going to have more to do. Let Amy to tell you more on Thursday. So we're going to do is, I would say, lab 15. The first one will be pathogenic modes. So first of all, we know fungi. This is eukaryotes. At different temperatures, they have a I will say it's a simply live form. At 25 degrees Celsius, it usually is a mode. And at 37 degrees Celsius, it is yeast. And we're going to focus on the modes today. So, in order to do that, I bought a bread which is very molded, which is terrible. I didn't wear a glove because I have to write on the blackboard. I have to do that, honestly. So it looks terrible. It's being put back in the trunk of my car for about 3 months. It looks like that. Very molded. You can look at the back, you can see it. At the back. And I also have this one, is also another bread, a different type of the bread, it's molded. But you see the color is different. This is like a white, this is like a yellowish, and a little bit like a greenish, grayish. And we also get a molds right here from our lab sample. So that's very good. We can see what it is. So what are the 3 major things for the molds? Right here in the picture. We basically have those 3, which is aspergillus. Aspergillus looks like and the second one is Leucopas. And what the Leucopas looks like that. And last one, everybody knows, it's penicillin. And penicillin, what it looks like. And you will see, usually the bottom like that. That's called mycelium. And you see it here, this is called hyphae, and it's spelling as and these like spores will be evaporated or spread into the air. In the Kentucky and Ohio area, or the Ohio River area, in the fall season, we usually, very common, some people will get a cough, which is from a fungi, and also a pathogenic mold called histoplasma mycosis. It's coming from the dropping of the birds. So I just want to let you know, the molded food, you not only you cannot eat it, you also cannot be exposed to the air, because those spores, the hyphae, or we call it a conidia, and on the top, they could be evaporated or spread into the air, and if you inhaled it, it cause a big problem. Some of them are carcinogenic chemicals. So be careful about the molds and the fungi. That's why I didn't bring these to the office and then bring here, and I just opened them right now. Okay. Now, there are different methods to testing pathogenic molds, but there is an easiest way. The easiest way to do is relatively very simple. Is you draw a big circle, same as what you did for the gram stick. But the difference is, we're not going to do any of the crystal violet or saffronin or those things. We're going to add a very special dye, a really blue one. That's called lactophenol cotton blue. And we're going to add one drop. So a big drop here. And then how we do it, we're going to using the tape. This tape is a little large, but it's okay. The tape, you're going to be holding like this to touch the molds. Let's say this area, you're going to touch like that. The mold is already there. And then on the slides, when you have a drop of the lactophenol cotton blue, you're going to stick to it. And you press it. Let the molds and the lactophenol cotton blue, they will be mixed with each other. And then you wait a little bit. We could be using 40x lens to do the observation. And you can find out, is that aspergillus, is that leucops, or is that penicillin? So that's a very interesting and a very easy study. Everybody could do it by yourself. So we're going to do this independently. Everybody do it. And how we do it? You're going to all come to the front desk to do it. So we will be rearrange it for you. You come to the front desk, we're going to do it for you. You could choose is from the plates, from my molded bread, and from my another molded bread. And I'll open it for you, and then you guys do it. Okay? You can do it one by one. So that's a very easy, quick test. Not yet, not done yet. The very quick, easy test to give you an idea what the fungi looks like. The next test is we have been talking about in the lecture, but we has not been practiced yet, which is called what's the terminology? It's called the Kirby-Beau test for antibiotics. So we're going to do a second trial. Is Kirby-Beau test. This Kirby-Beau test, what we're going to do right here, we will do 2 students as a pair. So there will be 1 plate. Okay, so how are we going to do it? You're going to get the agar plates. This agar looks very gentle or general. But this is not a neutron agar. This is called a Moeller-Helton agar. So Moeller-Helton agar is very special for antibiotics. And we will be, first of all, so it's in the broth. Okay, he already prepared the broth, so that's fine. You have an E. coli broth. The E. coli broth is like that. Now, we have a standards to measuring the turbidity of these broths. It's not saying you just put the bacteria, you can do the test. The broth looks like that, turns like relatively turbidity or cloudy. We call 0.5 mark Folland standard. What this means, around 10 to the power of 8 cells per ml. And ideally, you should pick from the plates, put in a 0.9% physiological salience and to do it, and compare to the silicone-made McFarland standards to see the turbidity. But Amy already did it for us. Okay, so we have this test tube. Then the second thing, what we're going to do, you're going to get an autoclaved cotton swab. But this is not going to do a throat culture. We're going to pick the cotton swab into the broth. And then we do a very general and very gentle perpendicular spread. And then we turn 90 degrees, we do a second spread. Which means we put the bacteria there. Then we're gonna, using this dispenser, we press a button to let the bacteria antibiotic containing disc to on the agar surface. This is very expensive, and I don't have so many for everybody. So we will do as a pair. And for you guys, we'll do nalidic acid and chlamydia. Other groups, they may do, I may have like sulfur drugs, carnamycin. This one will be nitrofurantoin. This is the one is the same thing. Okay, let's look at this one. What this one called? That's the same thing. This is the one is chlorophynicol. So we pick a couple of that. We do a demonstration. So once you do a dispenser, you should see there is a 2 discs. Usually they put a name there. Let's say P is penicillin, and then they put a number there. This P means it is penicillin. And what's the number there? That's concentration, usually microgram. Okay? Once we do that, this we will do 25 degrees Celsius, 48 hours. And also could be we do 35 degrees Celsius, 24 hours. And we can do next week. You will see there is more or less a transparent zone surrounded the disk. That is called inhibitory zone. Once the inhibitory zone has been formed, what we need to do? We're going to use a ruler to measure the diameter. The diameter is important because based on the diameter, we could have them into different categories. We're using I or just roll like this. We're using R represents resistant, S susceptible, and I is intermediate. Let's say we have a penicillin. We compare to a national standards. It should be 12 milliliter. This is a 14 milliliter. Let's say if our disk here, you get a number, is 15 milliliter, then compared to this table, what this means? That means this specific E. coli to penicillin is intermediate. If you get an inhibitory zone is 22 milliliter, that means this E. coli is susceptible to penicillin. And I mentioned on the lecture already, we using the terminology here, we call susceptible. We don't say sensitive. The reason is sensitive usually means the patient, the human patient, have allergic reaction for the antibiotics. So you know that using muscle injection or oral administration of the penicillin or the other antibiotics, you cannot just do it naturally. Lots of the time you have to do a skin test to see the allergic test because there is around like 0.5%, pretty high. A certain percentage of the population is allergic to antibiotics. If they are allergic to the antibiotics, we call it sensitive. So here, we call it susceptible. Okay? Now, if you say your inhibitory zone is only 10 milliliter, then what this means? It is resistant. That means this E. coli, if happened in this patient, you cannot even using the penicillin to do the treatment because it will cause resistance. And the resistance will be even severe if you repeat it to using the same dose, which means you are cultivating this resistant plasmid. Is that right? You're cultivating the all plasmid. Okay. So this is the work what we're gonna do today. I want everybody to work it to do the antibiotic resistant first, and then do the modes. And you and your bench partner could be doing the rotation. One person do the antibiotic resistant, another person is doing the pathogenic modes. And I'm not going to do a demonstration here for you because these are too nasty. I will be waiting for you a little bit for a couple of students to come in, and I will show them, then the TA can help. Because otherwise these modes goes everywhere, goes the whole bench. You walk, your hands all gonna have these modes. So wear the gloves and you come here, I'm gonna show you directly how we're gonna do it, okay?